And the most important thing that you will see is allergy, hypersensitivity. We talked about hypersensitivity type 1 last time. Okay? And today we're going to start from hypersensitivity 2, 3, and 4. There's a little bit of overlap, so we'll try to compare and contrast. And I'm going to start with the compare and contrast and then take you through the detail. Now, these are like my talking points, but if you want to listen to me in the YouTube, you can. So as I said, that Coombs and gel hypersensitivity classification of the allergy, right? So we have type 1, type 2, type 3, type 4. So there are 4 types of hypersensitivity reactions that you will get, further to what we discussed last week. This is what we discussed last time. And if you remember, we talked about it was IgE mediated, you pretty much know. If you remember, the IgE was to attach to the Fc portion of gamma C epsilon R1, and these were present on mast cell, basophil, and one more cell, eosinophils. You remember that? Okay, we did just talk about asthma. We talked about steroid and drugs and whatever. It was a little bit more details. Now, this is what I'm gonna start today. They look alike, but they are not, right? So this particular slide actually takes you to the concept. One of the concepts that you can see from here is that the immunology, if you remember immunity, we talked about immunity, and we talked about is going to be like cell-based or antibody-based. So some of the reactions are because of the cell, some of the antibodies. The antibodies we call it humoral, right? And cell we call cell-mediated. Right? One of the commonest questions we ask is that you can see it says cell, right? And then again, this is also cell-mediated. So some of them are cell-mediated, but having said that, antibodies also have a role. So these are the 2 antibodies, they have a role, right? And then over here, there is an IgM and IgG, very famous antibodies, right? This is the only one where apparently we don't see any antibody. That's why it's called, the other name for that is, we call it CMI, cell-mediated immunity, or we call DTH, delayed type hypersensitivity reaction. So these are the 2 terms that we normally use. Okay, so I'm gonna give you an idea how to separate. Forget type 1 because we already talked about that. You can revisit if you want to. In type 2, you can see both of them have cells which are going to lyse or cells which are going to kill, right? And they actually have another component which is complement. So there's another portion over here which is a protein. We call it complement mediated, right? And eventually all of this will cause cell death. All of this will cause cell death. So that we lose the function. Okay? Now keep that in your mind in a bigger picture, right? So it will be easier for you to figure out that the hypersensitive reaction is going to be one of these 4. Very rarely a little bit of overlap. Now let's go for this picture over here. So you can see this is an example of the type 2. Okay, so you can see from here, we have, did you miss something over here? What was this cell? What does this cell say?
Target cell.
Target cell, that's what I thought. Yeah, target cell, so something is missing. Target cell. Target cell. So that target cell has, an antibody attached to it. So you can see these are like antigens on the target cells. You can see multiple antigens on the target cells, right? And these antibodies come over here with the Fab portion of antibodies. They attach to these, all these different antigens, okay? With their fragment for antibody binding, and this is an FC segment, the legs and the arms, right? And then this one then is presented to a macrophage. So macrophage is an antigen presenting cell, or as the name suggests, macro means big eater. So this is the one that is going to phagocytose. Phagocytose. Take it in. Phagocytose. Okay? So this has a receptor, FC receptor, as we saw the FC receptors over here on the mast cells and the this is an FC receptor. This one was epsilon R1, very specific for mast cell, basophil, and eosinophil. But in this case, there is an FC receptor, it's just to hold on to your feet. So the antibodies will identify that, so they will connect over here, and this will be brought in. So this is like a brought in, okay? And in this specific case, what happens is that if you remember the structure of antibody, this structure of antibody, which has the fragment for antibody binding and FC portion, and they had this tiny little dots, if you remember, over here. Right? These dots over here are where the complement would bind. So this is a complement binding. So you can see the complement is going to bind. Complement is going to bind to these sites. These are the complement binding site. And especially complement, which is C3b. One of the components of C3b is one of the complements that binds to this, right? So the concept in this case, if you remember functions of antibodies, so I gave you at that time a mnemonic which says P-A-I-N, and I always forget the last one. What was the last one? P for precipitation, A for agglutination, what was I for?
The information.
Say it again?
The information.
I gave you a mnemonic for that. N is neutralization. And I for immobilization. Remember I gave you an example that there's a crime scene and but there's one I always miss. And I always ask my class to tell. What was the last one? C, complement activation. Complement activation. So these are the functions of antibody. These are their functions. You keep that in mind for this Friday exam. There are tons of questions of antibodies. I'm very fond of antibodies. So be prepared for questions for antibodies. So this receptor over here, which sees the complement, binds with that, and takes the whole complex in. It kind of takes it in. Okay. And the other important thing is that in this case, when you have an FC receptor, then you have these components of complement, right? So they form like a conglomeration, like a bullet, right? which is called membrane attack complex. So this membrane attack complex is like a hole. It bursts a hole into the cell and then causes osmotic lysis, because if it makes a hole into it, like a bullet, if it punctures that, then the equilibrium is lost. So intracellular and extracellular fluids balance will be lost, and then you will have a thing which is called osmotic lysis. This is also one form of a hypersensitivity reaction. Okay. And this one is like the membrane attack complex is like a, what do you call, a bullet, which is like a structure, like a barrel structure, right? So it's kind of punched into the cell, and osmosis will cause that. Okay? So far so good? The second important concept for this one, again, you have a target cell. You can see the target cell. So this target cell could be any cell. It could be a blood cell, it could be a joint cell, it could be a liver cell, any cell which is targeted, right? And it's going to initiate. And if you lose this target function, the disease will be accordingly. Like if it damages the liver cell, kidney cell, liver cell, whatever cells. Now in this case, there is a same target cell that you can see. It has all these antigens. You can see it has antigen. And in this case, it binds with an antibody. So there's an antibody over here. You can again, antibody, right? Antibody binds to these because antibody sees that, right? And this antibody, then the Fc portion of this antibody, which is the Fc portion, is seen by a cell, right, which is called natural killer cell. Natural killer cell. It sees that, okay, and this natural killer cell has this ability, is like SWAT or whatever, you know, more than the police. It carries a lot of weaponry, and this will going to attack this cell which is surrounded by antibody and kills the cell by a process that you already learned in DAS1, right? You did the DAS1, right? So this process is called apoptosis. Apoptosis. Right? So in this case, the cell is lysed by complement-mediated osmotic lysis. The damage is A type. In B type, it is antibody-dependent, cell-mediated cytotoxicity. It says in the book. Because the antibodies are there, right? And they bring the cells, and the cytotoxin means cell is killed. So you can see this cell is killed. So two type of damages that can occur in what? In the hypersensitivity reaction. So you can see from here, you know, this could be the target cell, can be your own cells, right? If they're your own cells, they're going to kill your own joint, your own self, because your own red blood cell, own cells, right? In the other form, we'll discuss in that. Let's discuss in this one. So in the other type is that again, you have this neuromuscular junction. This is a neuromuscular junction, if you remember that. Okay. So we have this synaptic cleft, and this is a motor neuron. Motor neuron has this sacs which releases acetylcholine. Okay. And then this acetylcholine comes over here. It binds to acetylcholine receptors on the other side. Okay. And then you have a normal neuromuscular junction, and which is called a contraction. So the contraction, right? Normally. But what happens is that for some reason, an antibody is formed by the cell against this acetylcholine receptor. So you have antibody to acetylcholine receptors. So what will happen is when acetylcholine will be released over here, right, and it has to come and bind with this receptor, it is already taken by the antibody. So what will happen? No contraction. Like myasthenia gravis, you have this drooping eyelid. We call something no contraction, paralysis, which will be, example is myasthenia gravis. Right? So this is also a damage, right? But this damage over here is antibody mediated damage that usually occurs where, it could be, and again, any antibody anywhere, but to give example, anti-receptor antibodies. So antibodies are coming over here, formed by the body, and block the normal function, leading to a disease. It's this myasthenia gravis over here, you see? That's a disease. Okay? Got it? Now, the difference between type 2 and type 3 is that in type 2, the cells pretty much, this antibody complex over here, this antigen antibody complex over here, right? This is in a way fixed. It's in the joint liver, it's a fixed. Right, so it's going to cause arthritis, it's going to cause carditis, so the whole situation is fixed. In type 3, if you pay attention to type 3, what happens in type 3 is when this immune complex reaction, right, between an antigen and an antibody is free to circulate. It picks up the antigen by the antibody, but it's free to circulate. And it circulates in the blood because it's a heavy molecule. It's going to either precipitate, agglutinate, and this will lead to damage, local damage, accumulate in the circulation and tissue damage. So it's going to go different parts of the body. Does that make sense? Now if you compare that with the fourth one, right, in this there is a cell, again cell-mediated CD4 T cell. CD4 T cell is there, and their cytokines are Th1 type, which are pro-inflammatory, right? And they release cytokines which cause local damage, and this stays for days and weeks. So if you compare this guy over here, right, the typical example is all of you were given this Monto test. You get tuberculin test, all of you is a mandatory tuberculin test. So what they do is, in the tuberculin test, right, they inject an antigen. So they inject this antigen, which is a TB antigen. So they inject the TB antigen in you on the skin. So this is skin. And skin is very powerful immune response, right? The idea is to test whether your T cells have a memory of TB. That's what they test. If it's negative, this means your T cells have never seen a TB before, so you're TB negative. But if you have TB, then their cells are there, they're going to react. So you will have within 3 days a reaction over here. But if you were born in any country other than US, then you should tell them that don't test me for this skin test, because you have had an injection in your when you were a kid, because they give an injection TB for protection. So then you will be tested for X-ray. It usually happens for the immigration. It's mandatory that nobody's going to give you a job unless you do the tuberculin test. But if you were born because you have been given a vaccine when you were a kid in all the countries except USA, which is called BCG, which is a TB vaccine. We don't know the efficacy for that, but many times people say ugly scar. It has an ugly scar for all those people who have had born other than US. In US, you're not supposed to give BCG. Okay? So you give this antigen comes, and this antigen is picked up the antigen presenting cell, and is going to be presented by MSC, right, to the T cells. You can see this is an antigen over here. It presented to a T cell, which is in this case a CD4 T cell, right? So if you remember, CD4 T cell develop either in Th1 type, in Th2 type. If it happens in Th1 type, this is going to be pro-inflammatory, and it's a Th17, which is a new one, right? And what happens is that this will lead to the reaction, inflammatory reaction. That's why they go, if you look at the monitor, they test like induration. They see like induration, redness, swelling, blah, 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 over time. So they go for 72 hours. It takes time. So in this case, you can imagine the antigen injected subcutaneously is processed by local antigen presenting cells like Langerhans cell in the skin. Remember, there was one assignment given to you, what are the names of APC in different part. In the skin, they're called Langerhans cells, right? Kupffer cells, there are different names for that. And then, this is what happens. So then this T cells recognize this peptide, which is given to, let's say, Monto's reaction as a TB, they give like a TB peptide, right, which is like the tuberculin peptide. And it is presented in MSC, we already the MSC, which is your gene, and differentiate into Th1 cell, we already know Th1 cell, and Th1 will produce cytokines. So in this case, the antigenic challenge activates T cell cytokines, and the cytokines will call in other cells, like macrophages and all, to come over here, and then they are going to react. So this means that your cells already know, it is there in your memory cell, that this particular antigen which is given to you is going to release all those cytokines like gamma interferon, TNF alpha, and they will cause a pro-inflammatory response. So neutrophils will go in the vessels, other cells will go, and then you will have edema, swelling, the classical signs of inflammation. So the typical example for type 4 reaction is cell-mediated, because cells are involved, T cells, CD4 T cells, and then there are cytokines involved which are pro-inflammatory, and there is a local damage that occurs in that particular area to lead to a typical response, which is a cell-mediated immune response. Okay? So you can see type 1, type 2, type 3, type 4, that's why it's called Coombs gel. Type 1 is straight, you can't miss it, because IgE mediated, mast cell, basal cell, you can easily do it. 2 and 3 are a little bit difficult, because in 2, again, there are antibodies involved and cells involved. The antibodies are IgG and IgM. And as I said, 2, most of the time in 2, the target cell is located like joints, and other places. And then again, it can, like for example, you may have it in liver, spleen, pancreas, anywhere. In type 3, the antigen is free, and antibody picks it up and it circulates. So it's like, we call it antigen-antibody complex. It's called immune complex. So it goes and it gets blocked in different area. Okay, so you may see a skin reaction, you may see a reaction anywhere. In type 4, it is a cell-mediated reaction. It takes a while, right? And the pro-inflammatory cytokines are involved, CD4 T cells involved. So many a time the questions usually for the final will be, you want to identify, isolate, which type of hypersensitivity has cells involved, which type of hypersensitivity has antibodies involved, and how do they work. And then again, if you look at the in your book, that I give examples of diseases, right? What are the diseases in type 1? What are the diseases in type 2? What are the diseases in type 3? And what are the diseases like contact dermatitis, tuberculin reaction, and nephritis. You have sometimes the immune complexes, they go and deposit themselves in kidneys, so they have kidney disease. Sometimes they go and deposit themselves in joints, you have joint disease. Sometimes they go and deposit in small vessels, you have small vascular diseases. So what are these triggers? They are antigen-antibody complexes. Got you? Any questions?